Dear students, today's class is about ultrastructure of nucleus. As we all know that nucleus is the controlling center of the cell. It is made up of nucleoplasm, chromosome and nucleolus and connected to some cell organelles such as endoplasmic reticulum and ribosomes. First, let me study about nucleoplasm. Nucleoplasm is a space between the nuclear envelope and nucleolus which is filled by transparent semi-solid granular and slightly acidophilic ground substances which is called as nucleoplasm. Along with this, chromatin thread and nucleolus remain suspended in it, that is nucleoplasm. Nucleoplasm contains mainly the nucleoprotein, along with this nucleic acid, proteins, enzymes and minerals. So this is about components of a nucleoplasm. The nucleoplasm is made up of nucleic acid such as DNA and RNA and some important proteins such as histones and some acidic proteins like phosphoproteins as well as some important enzymes such as DNA polymerase, RNA polymerase and phosphorylase and nucleosides. Some lipids too we can find inside the nucleoplasm. Along with this, some minerals are found inside the nucleoplasm. So this is about the composition of a nucleoplasm. Let me go for chromatin structure. The chromatin is the complex of the DNA, proteins, and RNA, which is called as a nucleic acid. And then these proteins are of two major classes. The first one is a basic, which is positively charged at neutral pH. They are stones, and second one is heterogeneous, which are largely acidic and negatively charged at neutral pH. So this is the group of protein collectively referred as a non-stone chromosomal proteins. And then these are the proteins which are commonly found inside the chromatin structure. Before the cell division, these chromatin threads corresponding to each individual chromosome are extended and dispersed within the nucleus. If you go to the higher order chromatin structure, actually the nuclear envelope helps to organize this chromatin by binding some parts of it to the inner nuclear envelope at sites that are closely associated with its nuclear pore. And then the chromatin becomes highly condensed during mitosis to form a compact metaphase chromosomes that are distributed to the daughter nucleus. Actually, transcribed genes are localized at the periphery of these territories adjacent to the channel which are separating the chromosomes. So this picture tells about the chromosomal organization, how the chromosome occupying a particular region inside the nucleus. The chromatin in interface nucleus is organized into or looped domains. These domain contains about 50 to 10 kb of DNA, and then they represent the functional unit inside the nucleus, which can independently regulate the gene expression. Okay. This picture tells about the first level of packaging, that is nucleosome inside the chromosome. Here you can see about seven small bead-like structure. Each bead-like structure here we are going to call it as a nucleosome, a complete nucleosome, each one. And inside the nucleosome you can see a octamer shape like structure which is made up of stones, and then there is a blue color thread like structure which is going to surround the nucleosome, that is called as DNA. And then the diameter of each nucleosome is about 11 nanometer, and then they are connected, each one is connected by linker DNA which has its own length varying about 8 to 114 nucleotide pairs. In the below you can see a complete nucleosome which is made up of octamer stones, and then you in the side you can see a green colored structure that is called as stone H1 and wrapped by DNA. So this is the first level of packaging of nucleosome inside the chromosome. Actually, in eukaryotic chromosome, one can see the three levels of DNA packaging. When we isolate a chromatin from the interface cells and we examine by electron microscope, one can find it consists of series of bead-like structure, which is already we have discussed in the last slide, that is the nucleosome, and then they are joined by a thin thread called DNA linkers, okay, or interbead, and then this bead is called as chromatin, that is nucleosome. In 1974, Korenberg he identified and described this basic structure unit of chromatin, that is nucleosome. This is clearly involves an octamer of stone molecules, and two each of stone H2A, H2B, H3 and H4 is involved inside the nucleosome, and then the binding of proteins to DNA in nucleosome protects the region of the DNA from the nuclease digestion. Nuclease is a type of enzyme which can digest the DNA, but the linker or interbead threads or linker DNA are they are susceptible to nuclease attack. So this picture clearly tells about what is found inside the chromosome. You can see in the picture a purple color which is called as a metaphase chromosome. Inside this you can see a chromatin material, chromatin thread which measures about 30 nanometer, and following that chromatin you can see a nucleosome having the diameter of 11 nanometer, and if you extend further you can see a DNA which is somehow greenish in color thread like structure which measures about 2 nanometer in the diameter. So this is how the chromosome is made up of, and then the chromatin materials are found inside the metaphase chromosome. Let me pass on to the next structure, nucleolus. In the picture you can see a darkly stained material in blue in color, that is called as a nucleus. So the nucleolus. The nucleolus is found inside the nucleus, which is highly darkly stained. It is the region where you can see the production of RNA. RNA production takes place. So, if you go to the nucleolus, it is the most prominent substructure within the nucleus, and then it was identified by Zerklin in 1953. He was the first one to see the nucleolus. It is the site of rRNA transcription and processing, and it is also helpful for ribosome assemblage. And then the nucleolus is a ribosome production factory and designed to fulfill the need for large scale production of rRNAs and assembly of ribosomal unit. Recent evidence suggests that the nucleolus also have more role in RNA modification, that are several types of RNA move in out of the nucleolus at specific stage during the cell cycle. Morphologically, nucleolus is made up of three distinct regions. Number one, fibrillar center, dense fibrillar component, and granular component. These three different regions are thought to represent the site of progressive stages of rRNA transcription, processing, and ribosome assembly. The rRNA genes are located in the fibrillar center, with transcription occurring primarily at the boundary of the fibrillar center and dense fibrillar component. Processing of rRNA is initiated in the dense fibrillar component and continues in the granular component, where the rRNA is assembled with the ribosomal protein to form nearly completed pre-ribosomal subunit ready for export to the cytoplasm. So this slide tells about the functions of a nucleolus, where the nucleolus is helpful in the formation of rRNA and transcription as well as processing of rRNA and the production of ribosomal rRNA ribosome subunits. Okay, and then the size of the nucleolus depends on the metabolic activity of the cell, and then the larger nucleolus is found in cell which are very active, especially during the protein synthesis. So this is another picture tells about the ribosome assembly which takes place inside the nucleus and nucleolus. How the ribosome proteins are entering inside the nucleolus, and then how they are assembled, and then they are modified, and then they sent outside towards the nucleus, nucleoplasm, and then out of nucleus they are going to settle inside the cytoplasm. This is the way of the formation of some important subunits, that is ribosomal subunits, which play very important role in the protein synthesis. If you summarize the difference between the nucleus and nucleolus, the nucleus is larger in size, nucleolus compared to the nucleolus, and then if you go for nucleus is bounded by nuclear envelope, but nucleolus doesn't bounded by any other membrane. There is no limiting membrane in the nucleolus, and then the nucleus contains some important material such as chromosome, and the nucleolus you can't see any chromosome inside the nucleolus. Nucleus is rich in DNA, whereas nucleolus is rich in RNA. So this is about the difference between the nucleus and nucleolus.